Hello and welcome everybody to today's webinar about structure-based design of riboswitch and kinase ligands. Our guest speaker today is Professor Ruth Brenk. Ruth obtained her PhD from the University of Marburg, where she was in Gerhard Klebe's group. And by now she's full professor at the University of Bergen in Norway. And now, without any further ado, let us welcome Ruth to give her presentation.
Compared to the properties of the library compounds, and we came up with some suggestions how to assemble screening libraries to get a good coverage of chemical space. But today I will talk to you about two different projects. So first I will talk about some RNA targets and then about some de novo design of kinase inhibitors. So why are we actually interested in RNA? As you might know, RNA is also an interesting drug target because RNA plays a key role in many biological processes. So it's particularly known, I guess, that RNA is very important for translating going from gene to proteins. But there's also other roles that RNA has in the body, and it can fold into diverse 3D structures very similar like proteins. And also like proteins, it can specifically recognize small molecules, and the most known target of RNA are probably antibiotics. So many antibiotics interact actually with RNAs and not with proteins. But a question in this RNA field is still, are the RNA binding sites actually trackable, or which types of ligands do they need to interact with an RNA? So some years ago in my group we developed a trackability prediction method called TrackBot, and then we wanted to apply this method now to RNA to find out how trackable RNA molecules actually are. Therefore I will first give you a short overview how TrackBot works and how we derived it. So we started with a data set of trackable and non-trackable proteins. Then had to locate the binding sites of the proteins, then use the binding site to calculate descriptors, and then we derived the model which we then could use to score the proteins. So how did we determine the binding site boundaries and the descriptors? So what we did there is we just docked a whole bunch of small molecule tracks into the binding sites, and for this docking we only use steric constraints. Then we keep the molecules with a good steric fit and can merge them into superligand. So this gives us the shape of the binding site, and then we can use this superligand to calculate the descriptors of the binding site. So what we used in the original TrackBot paper was that we used the hydrophobicity index of the amino acids in the binding site, relative occurrence of apolar amino acids, relative polar contact surface area, absolute apolar contact surface area, and the contact surface area. We then fed these descriptors in a PLS model so that we can actually do predictions. So how well does it work? Here's the results shown for the test sets. All the bars that are colored in blue come from proteins that were assigned to be trackable, and all the bars that are colored in orange come from proteins that are assigned to be less trackable. And then we can just order them by the score, like them here, which they get from our prediction method. And what you can see is that we overall get a very good separation between the less trackable and the trackable proteins, but in the middle we cannot be so sure. So if you just leave out this ambiguous region in the middle, is that we can very well predict trackable and non-trackable binding sites. Okay, now we wanted to apply this method for RNA. So what we had to do is we had to change a few descriptors, because as I said, some descriptors we used in the TrackBot method rely on the properties of the amino acids, and for sure in the RNA we have no amino acids, but we have the nucleotides, so we had to change the descriptors. So what we came up with is these descriptors here. So we kept the relative polar contact surface area, the absolute apolar contact surface area and the contact surface area. But we changed here this to the hydrophobicity index and the relative occurrence of apolar amino acids. So what we use when we apply the method to RNA is that we use the fraction of carbon atoms in the binding site and the difference of the solvent accessible surface area of the superligand when bound to the protein, or if the superligand would be free. So if we use this descriptors, we can then also train the PLS model again. And here's just an overview of the results. So if we have, and the results are just shown for the test set, so that the binding sets, they're not included in training the descriptors. So with the original trackpad method, we get these values here, and with the adapted method for RNA, we get very similar values. So by changing the descriptors, we are now able to apply also trackpad for RNA without losing a lot of the sensitivity and accuracy which we had from trackpad before. So which results do we get if we do this for RNA, or how does it look like? So we just assembled a lot of RNA crystal structures from the protein data bank, located the binding site, and used the trackpad method to score the binding sites. And the RNA structures are here sorted by the score they obtained from our method. And this bar here indicates, or this line, is the separation from the trackable to the less trackable binding sites. And what you can see is that there are indeed some RNA binding sites that score as very well trackable. And particularly, a lot of these binding sites here, which score trackable, are colored in orange. And in this scheme, it means orange is that they come from a certain RNA type of RNA, which is called riboswitches. So it appears that a lot of riboswitch binding sites are trackable. So if you want to design drugs against RNA, then at least from the binding site properties, the riboswitch seem to be extremely attractive. So what is a riboswitch? A riboswitch is able to regulate the gene activity on the level of RNA. So this is illustrated here in this schematic. So the riboswitch sits in the 5' untranslated region of the mRNA. And there's two types of riboswitch. There's on riboswitches and off riboswitches. And illustrated here is an off riboswitch. So a riboswitch can bind small metabolites. So in this case here, when the gene is translated in the on form, so it's switched on, then there's no metabolite bound, and we have this structure here of the RNA. When the metabolite is binding, the terminator loop, the aptamer domain of the riboswitch changes its conformation because the metabolite is binding to it, and as a consequence, the terminator loop is formed in the RNA, and the gene cannot be longer translated. So that's how an off riboswitch works. By binding a small metabolite to the RNA, we induce a change in the conformation of the RNA, and the gene's translation is switched off. There's also off riboswitch that just work the opposite. So if no metabolite is bound, then the gene translation is on. If the metabolite is binding, we get the change of RNA conformation again, but in this time then the gene translation is switched on. Where do these riboswitches occur? They are actually quite widespread in bacteria. About 2% of the genes in Bacillus subtilis are believed to be regulated by a riboswitch. And since these riboswitches are able to regulate the translation of essential genes, they're believed to be new drug targets for antibiotics. There's also a few crystal structures of riboswitches. So shown here on the left side is the crystals. The binding site was determined by crystallography in the of an adenine riboswitch. So we have the adenine bound to the riboswitch here. This is a rather small binding site, and then shown here on the right-hand side is the binding site of the TPP riboswitch, and there we have a larger binding site which has about the size of a typical protein binding site or enzyme binding site. So what we wanted to do in the beginning then is we wanted to use a simple RNA, the riboswitch binding site, in order to see if we can use this in principle for structure-based design, so if we can carry out a structure-based virtual screening against the binding site and come up with new ligands. And we used this adenine riboswitch binding site as a test case for this study. The reason for this was that the binding pocket is actually rather simple and small, so if we should get it right to dock against RNA, then we thought we should get it right in such a simple binding site. And there were also several ligands known that bind to the binding site and also compounds that do not bind, so we could actually test the predictions. And there were also 3 crystal structures in complex with different ligands, so we could also see if we could predict the binding modes correctly. Actually, predicting the binding modes turned out to be very easy. So here is a superposition of crystallography determined binding mode and the docked pose, and you can see almost no difference. So that was actually quite easy to dock these ligands in. And we could predict all 3 binding modes with an experimental error. Then we used the training database where we had compounds where we knew from literature sources that the compound should either bind or not bind into the adenine riboswitch binding site. So all the compounds that are used with black fonts are supposed, we know that their binding was confirmed experimentally, and all the compounds with red fonts, for these compounds binding could not be detected in experimental assays. So we docked all these compounds into the binding site and ordered them by score, and there we also get the very good separation between the binders at the top and the non-binders at the bottom of the list. And we see also 2 compounds where we had the false positive or false negative prediction. So this compound was predicted to bind but shouldn't bind, and this compound was predicted not to bind but should bind. So why did we not get them right? So for this adenine, the issue was predicting in the protonation states. So this is again the crystal structure of the riboswitching complex with adenine, and we presumably have a hydrogen bond here. And we also docked the adenine where we have a hydrogen bond attached here to this, hydrogen attached to this nitrogen. But actually it turns out that the pKa of this compound is so that it's probably actually deprotonated in solution. And if you have a negative charge here, then it makes a lot of sense why this compound cannot bind. So we got this compound wrong because we failed to predict the protonation state correctly. But it was nothing specific to RNA that failed. And then we had the N6-methyladenine, which we predicted not to bind, but it should bind. And the issue here is that that's actually the largest compound in the group, which has a methyl group here attached to this amino group. When we did the docking, we kept the water molecule here at this position, which was found in all 3 crystal structures that were published before we did the study. But when we actually got the crystal structure of this compound in complex with the riboswitch, we saw that this water molecule here is expelled, and that's also the reason why the docking didn't work. So in order to get this molecule docked correctly, we have to predict the water structure correctly, and I think that's still actually quite challenging. Okay, nevertheless, the docking worked very well, and then we proceeded to the more complex task, where we had the database with about 1,000 molecules or so that were small enough to sterically bind into the adenine riboswitch binding sites, and then we docked all the molecules into the binding site, ordered them by score, and purchased 5 ligands which were at the top of the list and which were not, where no studies had been carried out previously if they would bind or not. So these are the structures here. And actually 4 out of these 5 molecules turned out to bind to the riboswitch, so that was a very good and very nice result. And then we could also get the crystal structures of 3 of these compounds in complex with the riboswitch to see if we could also get the binding modes predicted correctly. So this is the structure of the first compound, very nice electron density, and it matches perfectly well the crystallographically determined binding mode matches perfectly well with the dock binding mode. This is the second compound. Here we had again issues with the water structure, so we ran I mean, it's a symmetrical compound, so you can only interfere from the hydrogen bonding interactions where the methyl group is and where the amino group is. But we believe that the amino group is here, whereas in docking we predicted the other way around, and we believe that this also has to do with the water molecule, which can actually shift position, and we didn't consider this during docking. Then finally, the third compound, there we saw two different binding modes in the crystal structure, and one of them was the one we predicted by docking. So overall, I think the docking to the RNA structure worked very well. We were able to reproduce the previously published results, and we could also predict new ligands, and it's I think always important if with new ligands that some of them were based upon scaffolds that were not previously known to bind to adenine-sending riboswitches. So docking was really able to give you some new chemical structures there. And interestingly, none of the problems that we encountered in the study were specific to RNA-ligand docking. So what we thought the issues was, since we use DOCK, which has a force field-based scoring function, that we have issues with the balance between the electrostatic energy and the van der Waals energy, because the RNA structures can be highly charged. But that doesn't seem to be the case. But the issues we had are issues I think we're also familiar with protein-ligand docking, like multiple binding modes, water molecules, and protonation states. But I think overall, the study worked very well, and we are very confident to take this RNA-ligand docking forward to more complex targets, which actually will be really interesting as targets for new antibiotics. And one of these targets we're currently working on is the FNM riboswitch. This riboswitch has already been demonstrated to be a potential target for antibiotics. So one thing is here, this is riboflavin. This is believed to be the natural ligand of the FNM riboswitch. There is a natural product called riboflavin, which is chemically very much related to riboflavin. So the only difference here is this group. And riboflavin has been shown to have antibiotic activity, and it could be shown by mutation studies that riboflavin most likely acts in bacteria via binding to the FMN riboswitch. And then last year, people from Merck published another compound which they named ribosil. And this was discovered by phenotypic screens looking for new antibiotics, and later they found that the target for ribosil is actually also the FNM riboswitch. People at Merck could also determine the crystal structure of ribosilin complex with the FMN riboswitch. So an overlay is shown here. So the green carbon atoms come from the FMN, and the blue carbon atoms come from the ribosil. So if you superimpose the compounds, then you can actually see that the binding modes are quite different, which the ligands adapt, because the FMN is here forming direct interactions with this adenine, whereas the ribosil is pushed a bit further into the binding site. Now we did the virtual screen against the riboswitch and came up with a small fragment-like compound where we could determine the binding affinity by ITC experiments. And we are currently trying to get the crystal structure of our small molecule in complex with the mRNA riboswitch, and also to synthesize some derivatives to optimize the affinity of the compound. Okay, this was the part of with the RNA. So we have adapted our trackability method, a trackpad, that we can also use it to RNA structures. There are indeed some RNA structures deposited in the PDB which have binding sites that appear to be trackable. And a large group of these RNA molecules with trackable binding sites are riboswitches. And we could demonstrate that the docking against these riboswitches can actually work very well. And we also discovered a new ligand for the FNM riboswitch, which is a potential target for antibiotics. Okay, I think the RNA is probably a bit less known or less established targets in drug discovery, whereas the kinases are one of the most studied targets, I would guess, at the moment. So the second part of my talk is now about the novo design of protein kinase inhibitors. I believe I don't have to tell you much about protein kinases. They're very important enzymes in the human body. They're a drug target for a range of diseases. The challenges in the field is to reach selectivity, because a lot of the compounds bind into the ATP binding site. and also to come up with new chemical matters, which is where you don't run into pattern issues if you want to optimize the compound. And with this in mind, we wanted to develop molecules which have new cores which were not reported to be active against kinases before. So how did we do this? Here's a schematic view of the ATP binding site in the kinase. This is where the ATP will bind. Here's the hinge region. So we were not interested in compounds that cannot interact with the hinge region. So we were targeting the classical adenine binding region in our approach. We were also not interested in compounds that can interact with the hinge, but where already kinase activity had been published. But what we were looking for is compounds that could potentially interact with the hinge, but have not been reported previously to be kinase active, and that could also be elaborated to reach the cytophobic region 1 or 2 in order to increase affinity and selectivity. So how did we do this? So we started with a pool of commercially available compounds. At that time we had a database with about 2.3 million molecules in my group, and then we extracted what we call core fragments. So how do we do this? This is a very hypothetical protein, which we then take apart to extract the core fragment, and we define the core fragment as a ring system plus the directly attached polar functional groups. So if we would dissect this compound into a core fragment, we end up with these ring systems plus these polar groups attached to it. Okay, we did this with all these 2.3 million compounds, which gave us about 84,000 unique core fragments. Then we selected the core fragments of interest. So we did the pharmacophore. I think that's also quite established, right? So we wanted to interact with the hinge region, so we needed a certain donor-acceptor-donor motif, and we also need an aromatic ring in the right distance. And we looked also only for core fragments that were in the fragment size range, so that we could further elaborate into larger compounds. So this left us with about 6,000 fragments, and then we docked these core fragments against a panel of kinases. So how did we choose this panel of kinases? This work was carried out when I was in Dundee, and there my colleagues in the protein phosphorylation unit, they have an assay panel. At that time I think it was about 120 different kinases for which they have kinase assays running, and we just took the crystal structures of all the kinases we could get hold of that are part of this panel. So then we docked the kinase in all these crystal structures and ranked the fragments by score. So this is the matrix we get. We had about 40 to 60 different protein kinase structures on this axis, and then we had the different docked core fragments here. And the ones that are marked in green as opposed to bind strongly to a kinase, yellow middle, red weak, and black they predicted not to bind. And then at this stage of the study we were looking for core fragments that are able to bind to a whole range of different kinases. So if we blow up this top 100 region of the non-selected core fragments, that's what we ended up with. And it turned out that actually 73% of the core fragments were already known to bind to protein kinases. So this gave us a lot of confidence in our method that we can actually predict the right fragments. And the remaining 27 fragments were novel, so they had no reported kinase activity. We could also compare the binding modes of the core fragments we obtained with crystal structures with protein kinase complexes, which contained similar fragments. So in green is always shown the dot binding mode, and with orange carbons the crystal structure. You could see that for these core fragments we got also the binding modes predicted very well. Okay, then we had to filter them and had to shortlist which of the fragments we actually wanted to work with, and then come up with a synthetic route to synthesize and to test them. So how did we select the fragments we worked with? So they had to have a promising binding mode, so they had to interact with the hinge region in a certain pattern. They had to have a good score against many different kinases. So they should be novel, so they should not have reported kinase activity in either Campbell or SciFinder databases. And they should have suitable synthetic points to introduce diversity, and they should be synthetic feasible. So we ended up with 6 different core fragments. And this is the different core fragments we decided to work with. And this is the predicted binding modes then relative to the hinge region shown in gray. And then we synthesized, or the students synthesized small libraries around the 6 core fragments, and in total we ended up with about 200 compounds. And then for a proof of concept study, we selected 15 different compounds out of these 200 and tested them at the concentration of 100 micromolar against a panel of 117 different kinases. So the 15 compounds we worked with are shown here. In blue is always the core structure, and then in black is the attached substituents. And even though the core structure was fragment-like, the full compounds were still rather small and in the fragment-like space. And that was also the reason why we decided to test them at rather high concentration. So this is the result from a compound-centric view. In green is, oh now I have to cheat and have to check this. What green was. So if the yellow bars is at least 40% inhibition at 100 micromolar, and the green bars is at least 70% inhibition at 100 micromolar. So you can see at least at the lower concentration, all the 15 compounds we tested were active against at least one kinase. So on this axis we have the different 15 compounds. On this axis we have the number of kinases that were inhibited. So some compounds were rather specific and inhibited only a small number of kinases, and some compounds were rather unspecific and inhibited a large number of kinases. So that's a compound-centric view. Then we can also look at the results from a kinase-centric view. So here we show how many hits a particular kinase obtained, by how many compounds of kinase were inhibited, and for how many compounds this was the case. And again, yellow is 40% inhibition at 100 micromolar, and green is 70%, at least 70% inhibition. So at 40% inhibition, there were here about 25 kinases that were not inhibited by a single compound, and if we go to the higher cutoff, then we had about 75 kinases out of this 117 that were not inhibited by a single compound. But then we also had kinases that were inhibited by a large number of compounds. So also for kinases, some seem to be easier to inhibit as others, and this has also been published in other studies before. But interesting was also that the kinases that appear to be easily inhibited by us were reported to be more difficult to inhibit by others, and vice versa. So I think if you say a kinase is easy to inhibit or difficult, you can only say this relative to the compounds that you are using. Okay, then we also took some compounds forward for hit confirmation, where we determined IC50s against different kinases. This is shown here. So some compounds were rather weak, and some compounds were in the low micromolar range. If you look at the this is 50 down. If you look at the ligand efficiency, so this bar, this dotted line here is the cutoff of 0.3 ligand efficiency, and then you can see that quite a few of the compounds were also quite efficient. So that worked in principle out quite well again. So we could design compounds that have a new core that was not active to be predicted and not known to be active against kinases before, but they turned out to be active at kinase, and actually some of them bound actually also quite potently to them. We also got the crystal structure of one of these compounds in complex with CSAC, and the crystallography determined binding mode also matched the ones that we had predicted from docking. But so how could we also predict the selectivity by docking? So that's shown on this slide, and that might look very busy to you, but you don't have to be able to read the numbers. You just have to get the general picture overview indicated by the colors. So what's plotted here? So on this axis here, we have all the different kinases for which we had the crystal structure, and we carried out docking. And here, on this axis, we have all the different compounds. And then the top line for each compound is always the experimental results from the assay. So if the compound was active in the assay, then the field is colored with orange. If the compound was active in docking or got a good docking score, then the field is colored in blue. So in an ideal world, if we could predict the selectivity of the compounds, we should always have an orange field over a light blue field. And you can easily see that this is not the case. So while it was relatively easy to come up with compounds that are active for kinases, where we completely failed was to predict for which kinase the compound would be active. So I think there is a lot of work to do still for us academics to fine-tune the scoring function and to really that we can get more specific with our predictions. So that brings me already at the end of the talk. For sure, I did not carry out the work myself. So the people I have to thank to are Agata Kosowski and Daniel Mythas and Orincha Sarkar, who worked on the track predictions, and that was a collaboration with Stefan Schmidt from AstraZeneca. The work on the adenine riboswitch was carried out by Peter Donaldhope, Colin Hammond, David Lillian, in the collaboration with Rob Beatty at the University of Colorado, and Thomas Wehler, who is on the photo here. He carried out the FMN riboswitch docking, and then Robert Ulrich did the design and synthesis of the kinase ligands, and this was a collaboration with MSD at the time. So thank you very much for your attention.